Thank you, Craig, for the humorous yet kind introduction. Just for the record, my name contains 66.66% vowels, if you care to count them. So, but it's not about vowels that I would like to talk to you today. And it is about a certain aspect of our research that involves the understanding of how DNA molecules move locally and globally to perform their amazing biological activity. Before I talk about that, because this is a larger departmental talk, I thought to present first what are we interested in general, the tools that we use, and the kind of techniques that we employ. So in the most general sense, we're interested in the description of biomolecular dynamics. So consider an Erlenmeyer flask like this one with on the order of 10 to the 23 molecules inside it. We would like to understand how one or a few molecules in there move at the atomic level. So in order to do that, we achieve what I teach in thermodynamics and statistical mechanics at the graduate level, and I'm happy to see that some of the students in those classes are here. We basically offer a statistical mechanical picture, a microscopic picture of the macroscopic thermodynamics of solutions. And the chemical systems of interest here are rather complex. They involve a large number of atoms that interact through covalent and non-covalent interactions, and these interactions takes place at a wide distributions of time, length, and energy scales, which makes the problem at hand not too easy. And the fact that it's a difficult question, so the problem is relatively difficult enough to warrant the award last year of the Nobel Prize in Chemistry for the development of computational techniques that address the multi-scale nature of the chemical systems involved in such instances, in biomolecular instances. So, okay, so this is what we're interested in. What do we actually do in order to simulate the dynamics? We don't do much more than basically integrate Newton's equations of motion in time. This involves calculating the force on all the atoms in the molecule. So you saw here a number of atoms wiggling and jiggling. And why do they wiggle and jiggle? Because there's a force on each of them, and the force on each of them comes from the gradient of a potential. What potential? It's an empirical potential. It's the potential for which the Nobel Prize has been awarded. And it involves very simple considerations. When you have two atoms that are covalently bonded, right, through a covalent chemical bond, we describe this as putting a spring between the two atoms, right? So if you squeeze the spring, the energy goes up. If you extend the spring, the energy will also go up, and it does so harmonically. So we have a potential energy term that's harmonic in the bonds. When we have an angle that's formed by three atoms that are pairwise, this with this and this with the other one connected covalently, right? The angle can fluctuate. And if you squeeze the angle, the energy will go up. And if you extend the angle, it'll also go up harmonically. So we have a harmonic angle term. If we rotate the leftmost atom and the rightmost atom around the bond created by the central two atoms, between the central two atoms, this is called a dihedral angle. You can almost see it. It's the angle between the plane passing through these three atoms and the plane passing through these three atoms. These two planes make a dihedral, and that dihedral has a potential energy associated with it that oscillates in a first approximation periodically. So that's another term in the potential. Two atoms can also interact if they are necessarily directly related by bonds and angles through space, and that interaction is either modeled as a van der Waals interaction, or as an electrostatic interaction where each atom has a little charge on it that, again, is calculated as a parameter of this otherwise empirical model. We call this an empirical model because it is not a solution of the Schrödinger equation, right? We fit these constants, the spring constant of the bond, the spring constant and the angle, the curvature in the bottom and the tops of the harmonic dihedral potential, the A's and B's in the Lennard-Jones interactions describing van der Waals attraction and repulsion, and the charges Q1 and Q2 of two atoms that interact via electrostatic interaction. All these parameters come from either experiment or a more careful quantum mechanical calculation. But at the end of the day, for each atom in the simulation, we have a force, right? So that force is going to move the atom, and it's going to move it according to Newton's equation of motion. It'll move it such that the time derivative of the time derivative of the velocity, which is the acceleration, will change in a manner that's proportional to the force, and from the velocity being the time derivative of the position, you can calculate position. So we basically, by integrating, by solving this Newton equation of motion, we get the position and the velocity of all the atoms at all those times. And those positions are basically shown here. They are a time sequence of events. They are making a molecular movie. The atom is at a different position at each time. Where does that position come from? The solution of the Newtonian equations of motion. So you may say, well, this is relatively trivial. It's a simple classical equation of motion. We just need to solve it, and then we'll get everything. But how exactly do you get everything, and what does everything mean? Well, we're interested in thermodynamics, not per se in the dynamics of the molecule, and we're interested in the thermodynamics by definition. This means for 10 to the 23 molecules, but we've only simulated one. So in order to extract the thermodynamics from the microscopic simulation of one temperature, we have to be able to use the tools of statistical mechanics to relate a microscopic picture, a time-dependent picture, to an ensemble picture. But even that is not sufficient, because when you actually integrate the equations of motion, and the equations of motions are integrated not on a laptop such as this one, but on many CPUs that work in parallel and communicate to each other. These are called large-scale parallel computing. So each CPU calculates the positions and the forces of some of the system and communicates with each other to pass on the forces, velocities, and positions. And in the end, we're able to solve problems. We're able to show movies on the time scale, say, of microseconds of the real life of that molecule. And that takes us in excess of when we calculate all the systems that we do and everything that we're interested in, we use of the order of 100,000 CPU days per year, which is compared with the number of days of brain, the number of brain days we maximally can use a year. So my students should be attentive. In 2 years, we'll be able to have an extra day, right, as we did in 2012, because it's a leap year. So on the development side, I should say that half of our work is using just pen and papers and erasers. Erasers are important. The reason we need that is because I said we can make these movies on the microsecond time scale, okay? But very important molecular biology, as some of you know, takes place on the millisecond to second time scale. And that we cannot simulate directly, so we need new methods that extend the reach of the kinetical and relaxation properties that we can simulate in the computer. However, today I won't talk about the theoretical methods development. Instead, what I will do is to walk you through a zoo of systems that undergo what is, to me, exciting molecular gymnastics that involves deoxyribonucleic acids, DNA molecules. So, and we will do this at the local scale first, and then we will talk about global such conformational transitions. But why are we interested in DNA gymnastics to start with? Well, according to the textbook basics of the DNA descriptions, we have 4 kinds of bases, A, T, C, G, and we have unique ways of pairing A and T and C and G. Although what exactly we define by unique, we will see later. So the canonical picture of DNA, the one you always see in molecular biology textbooks, is that of a perfect double helical strand with A pairing with T always like this, and C pairing with G always like this. So you have 2 hydrogen bonds here and 3 hydrogen bonds. And that's fundamentally the description of DNA on the local scale. But on the global scale, DNA is much more exciting. It behaves very much like a telephone cord, right? If you think that your telephone cord has 2 wires in it, they are the 2 strands, let's say, and you take that telephone cord and you twist one end relative to the other, what you will see is that you produce what are called supercoils. So in addition to the coil of the telephone cord, you have a coil of coils. The helical axis of the coil coils around itself. So you have then supercoils like this. Moreover, you have very complex squeezed geometries of DNA. And we're interested in all these contorted states for 2 reasons. First is a fundamental molecular biology reason. For example, DNA in genes is wrapped around proteins that are called nucleosomes. And they are very tightly wrapped, much tighter wrapped than it is possible to bend the DNA without the protein. Moreover, if a certain gene is to be repressed, you want to suppress the expression of a certain gene. You don't want to make a certain kind of protein. Well, the DNA that codes for that protein is looped. So if you loop it, then you repress the expression of that gene intervening between the 2 points that touch each other when you loop the DNA. So it's a very fundamental genetic transaction. Moreover, DNA, the 2 strands of the DNA, are unzipped. One is peeled off from the other, so the hydrogen bonds are destroyed during the process of DNA replication, right, where one strand of the DNA serves as a template for the replication of new DNA. Even furthermore, DNA is tightly squeezed into the capsids of viruses at a pressure that exceeds 10 times the pressure of a champagne bottle. So the geometries that occur there are very far away from the canonical textbook picture of double-stranded DNA. So when we think of DNA, we should think, at least at a global scale, as being more like these nautical knots that you see here, rather than the straight situation presented in typical introductory descriptions. There are also exciting new applications in the realm of biotechnology. For example, we can design nanomechanical devices. We can use the energy stored in the supercoils, right? Remember I said that you take a telephone cord and you supercoil. There's energy stored there, and similarly you can store energy, and perhaps you can make DNA nanopropellers that will push themselves through the solution of a biological or nanotechnological application to target a specific region in that application. Moreover, we can make, not we as a community, in general, it is possible to make nanopores out of DNA that mimic the nanopores that are made by proteins. These are hollow, roughly cylinders that penetrate the lipid bilayer of cells and serve as an opportunity to shove things around through the hole, for example, to perform individualized gene therapy. You can push DNA inside the cell through the bilayer lipid of membranes. So there are exciting opportunities and challenges at the same time in studying the conformational transitions of contorted DNA. And the plan of the talk is to go through these opportunities and challenges, and starting at the local DNA level, we will see why is it important to understand the way in which bases move relative to each other in the double helix. Then I will talk about the removal of the supercoils of DNA, how is that catalyzed by enzymes called topoisomerases, and then go into the realm of nanotechnology and talk about how dendrimers condense DNA and what are the forces, the characteristic forces and processes at play. And lastly, I will talk about the way in which DNA gets out and goes into the viral capsids to undergo viral import and ejection. So let's start with the story. We will discuss the local DNA gymnastics involving flipping out DNA bases. In the 50s, at King's College, Rosalind Franklin started to produce really detailed X-ray photographic pictures of DNA. And Linus Pauling, who successfully predicted the geometry of an alpha helix, so not of a DNA, but an alpha helix, took a crack at it and suggested that the pictures that he has not yet seen of Rosalind Franklin, but the data that existed up to that point, would indicate that the DNA is such that it has 3 helices, but the sugar phosphate backbone of the helix is inside, and these 4 things, A, T, C, Gs, the bases are out. While Watson and Crick, being local, have seen the pictures coming out of King's College, and they were able to come up with the real model for double helical DNA. That model of Watson and Crick shows that there are these 2 backbones made of sugars and phosphates repeating periodically, and the DNA bases are inside, and they hydrogen bond with each other on the inside, not on the outside, as Pauling has suggested, having in mind his successful model of an alpha helix. In an alpha helix, the situation is opposite. You have the backbone of the protein is inside, and the residues, the amino acid residues, are sticking out into the solution. Right, so Pauling's model was wrong, and the paper went into the Hall of Shame of theoretical molecular biology, or so it seems. Because what happened in by the way, tracing back, the story is much more exciting than I told it, the Pauling versus Watson and Crick. And so I recommend that you read on it. The novel experiments involved are of the single molecule type, and they involve tweezing the DNA at the single molecule level. And they suggest that there is an abrupt transition in which DNA suddenly elongates by 70% of what it is in the so-called bDNA state. This is roughly the Watson and Crick model. So why is that? First of all, what are these experiments? These experiments employ either magnetic beads, as shown here. You have a magnetic bead that attaches to a double-stranded DNA. This is just not one strand, it's double-stranded DNA. And because it's magnetic, you can actually twist it. So there is a torque that you can induce on the DNA, and at the same time you can pull the magnets up and you can apply tension, right? So I have a piece of thread and I can apply tension at its ends, and I can twist around it and I can apply torque with these magnetic tweezers. Another kind, this was by Ben-Simon and Croquette in Paris. A similar experiment, but involving optical tweezers, was performed by Carlos Bustamante and his collaborators at Berkeley. But the idea is the same, to twist and pull DNA. So what we set to do was to analyze in detail what kind of new DNA is seen in the experiment. Why do these experiments see a sudden jolt of extension in DNA that extends it by 70%? And moreover, why is it that they see that when they add to the solution moieties that chop the bases off, they do that. They lose, the DNA loses its bases. So what we set to do was a molecular dynamics simulation. We used those supercomputers that you've seen a couple of slides ago, and we pulled and torqued DNA virtually. So we created a model in which we stacked an initial B-form DNA, and we pulled these two strands up and these two strands down, and we also applied a torque at the end, at this end and at that end. As you can see, there's a transition from a B-DNA state to something that looks like Pauling's original model, where the sugar phosphate backbone is inside and the bases are out. So we can be a little bit more quantitative than that and calculate a free energy profile as a function of a reaction coordinate that goes from B-DNA, from Watson-Crick DNA, to Pauling DNA. And this free energy profile, the derivative of this free energy profile, will give us the force and the torque on the DNA exactly. So then we can go ahead and compute those tensions and torques from the simulation. They're showed here. And then we can go and check with existing experimental data. And what you see here is an experimentally calibrated force versus torque phase diagram. Okay? So this, if you want to think in terms of thermodynamics, this could be like a Clausius-Clapeyron phase diagram, where you see a solid, liquid, and vapor, right, for example. Anyway, here, the bottom line is that the calculations, which are showed in red, actually go very well through the phase diagram that predicts separation between a B-like DNA, Watson-Crick, and the P-like DNA. And we see that the transition occurs very close to a critical point, which is the minimum point below which the transition cannot go. So that was a fairly rewarding application of a computer exercise in calculating the potential of mean force. The simulations were in good accord with the experiments that were performed on the same kind of molecules. Please interrupt me with questions anytime. Okay, so that was flipping over. What happened was that the DNA base pairs, they were hydrogen bonded with each other, and then because I twist and pull, it's only natural, you'll think, right, that the DNA flips out, and that's basically confirmed by the simulation. But now we want to see another instance of flipping, this time not flipping out, but flipping over. So a base is does not have mirror symmetry, just like my hand. It's one way to present you this side, right, of my hand, and that's another situation if I flip the base over, right? There's a question before we talk about flipping over.
The P state of the DNA is like a natural state.
P state?
I know that there is some changing, for example, from the B DNA to A DNA depending on the hydration.
Yeah, that's a great question. So to preface it, let's go here. There are various DNA forms. This Watson-Crick base paired, but they can behave differently. They can, from the top view, they can look like this, or they can look like this, or they can even look in a way in which the right-handed coil is left-handed. That's called Z-DNA. And all these conditions are physiological, can be physiological, and they are mediated either by proteins that bind to the DNA or by a change in the solution surrounding DNA. Here, pDNA does not exist, so it's not a total vindication of Pauling. It's not that I will ever see a pDNA situation like this in solution. It only appears, it has some high free energy, But that is lowered only when you apply forces and torque. So only in a situation where you pull and twist DNA can you stabilize this form. So again, Pauling's vindication, well, partially because of this. Yes, so it turns out that because I was a postdoctoral student of Martin Karplus, the Nobel Prize winner of last year, and Karplus was a graduate student of Pauling, my student at Michigan, Jeff Vereshchinsky, who is now at the Illinois Institute of Technology, was very happy that he was able to vindicate his great-grandfather, scientifically speaking. But that, again, it's only a partial vindication. For at least another reason, Pauling's model had 3 helices of DNA, not 2. So 3 strands of DNA, not 2, in one helix. Okay. So, so that was a study on the flip over of a flip out of DNA bases. But what happens to DNA when you flip it out? So, as I said, A is binding with T, and they're binding through these hydrogen bonds. But and C involves 3 hydrogen bonds. So let's make this akin to the way in which these 2 ballet dancers hold on to each other. They grip at 3 points, right? And in principle, but notice the pose of the ballerina. She is facing towards you. There's another possibility in which this time only 2 hydrogen bonds are created, but this time the ballerina is facing the back. So her back is facing you. This involves a flip over. Okay? Moreover, note that the right hand of this performer grabs the right hand of this performer, but here his right hand grabs her left hand. So there is a change in the acceptor-donor pair that hydrogen bonds. In other words, it involves a flip over, but at the same time a rearrangement of the acceptor and donor positions of the 2 bases. In a closer look, and that alternative hydrogen bonding pattern is called the Hoogsteen pattern, after the scientist who looked at this problem. So, for example, in a Watson-Crick situation, shown here in the upper left side, you have an AT double, two hydrogen bonds, but then in a Hoogsteen base pair, you see that this 5-member ring actually flips over, and you produce a new hydrogen bond that didn't exist before. Similarly here, you have 3 hydrogen bonds. 1 is maintained and 2 other, and a new one, 2 are destroyed, and a new one is created. Okay? This is just a simplified view. There are many more situations that you can create because you have more possibilities for acceptor-donor pairs. But this is the simplest situation, and it is exciting because the existence of Hoogsteen base pairs is not news. It has been predicted and seen many times before, but it has always been seen in contexts in which a protein binds DNA and deforms it severely. The Hoogsteen base pair has never been seen in what we call naked DNA, which means DNA in the solution in the absence of protein. Not been seen until very recently, when my collaborator, Hashim al-Hashimi, then at the University of Michigan and now at Duke University, has seen in a liquid state NMR experiment the existence of a very low populated, 1 in 100 relative to the Watson-Crick, a very low populated Hoogsteen population that lived on the millisecond time scale. And so what we set to do in collaboration with him was to create a movie, a transition between the Watson-Crick and the Hoogsteen. And you see it here in its details of hydrogen bonding. Okay, so we flip the base over, back and forth between Watson-Crick and Hoogsteen. Okay, quiz. Can you tell me if it's an AT or a CG? Look at the initial and the final number of hydrogen bonds. There are 2 for each, right? So it's an AT base pair. Okay, so these are the kind of things that we can do. We can study the thermodynamics and the transitions between local properties of DNA that are important either for understanding novel single molecule experiments or for understanding the thermodynamics and kinetics of liquid state NMR populations. Next, I will talk about 3 more things. So I will walk you through the zoo and show you 3 more animals. First will be topoisomerase interacting with DNA and removing its supercoils. Secondly, I will talk about man-made dendrimers interacting with DNA. So this will involve calculations of how these dendrimers compact DNA. And lastly, I will talk about DNA inside a virus. That would be how much time left?
25 minutes.
25, okay. So this is an actual picture of the supercoiling state of DNA. Okay, so remember with the telephone cord analogy in mind, this is not supercoiled. And here, it's more and more supercoiled. So imagine that you have the supercoiled and circular DNA. We have to keep it circular. We have to talk about it being circular. Not necessarily. You can also talk about DNA that's not circular, but its ends are relatively fixed relative to the motion of the supercoil between those 2 ends. So how can you obtain from a supercoiled DNA non-supercoiled DNA unless you do something to your DNA? You cannot really, right? So imagine you have 2 rubber bands and you twist them one around the other. First they will make a coil. One will coil around the other. Then you keep coiling. Then you have a coil of coils. So you have a supercoil, right? And now imagine that you put together the 2 ends on which you grabbed, right? You have circular DNA. No matter what you do to the DNA, you cannot reduce its so-called linking number, the number of times the DNA crosses over itself, and plus the number of times the helical axis crosses over itself is what's called the topological invariant. No matter what you do to a topologically sound DNA, you cannot do it. But if you nick DNA, so imagine that you come with scissors and you nick not both, but one of the strands, right, of the rubber bands, what will happen with the nicked strand is that it will rotate around the other and it will release supercoils. Well, that's exactly what topoisomerases do. The binding involves the covalent formation of a bond between the sugar phosphate backbone and the protein, and the cut DNA downstream of the cut rotates, and the number of times it rotates is the number of supercoils it releases. So the situation is pretty much like this movie produced by my collaborator Nienke Dekker at Delft University at that time. All my collaborators seem to move, including myself. So the movie here shows the Pac-Man-like enzyme, the blue thing, that binds to double-stranded DNA that supercoils. So you see here the supercoils are downstream. So what happens is, first of all, there's a cut of one strand, and the energy stored in the DNA serves as the source to produce the rotation of the cut duplex around, pivoting around the intact one. Okay? So that's the idea. How do we know that this is so, and moreover, how can we measure it? Before I show you how this was actually measured and how real-time and how the kinetics of this process was obtained in real time, let me tell you that we can actually simulate this because there exist crystal structures of the protein in contact with the DNA. So In this particular work, which was done with Jeff Vereschinsky and my former postdoc Leven Sari, who is now associate dean at Fatih University in Istanbul, was a computer simulation in which we placed double-stranded DNA inside the grab of the Pac-Man-like enzyme. So here you see the enzyme this time is in green, with some of its domains colored in red, and the DNA is in yellow. Now, what we do is to calculate the potential of mean force for this rotation. It's basically a free energy profile along the rotational degree of freedom. The quantities needed are recorded, and we can calculate the potential of mean force. So if we do that then what is this? Well, this is the same thing, but this time, if you notice, the DNA rotates in the other direction. The reason is because along the helical axis, DNA has broken rotational symmetry. So it's one way to twist the DNA along its grain. It will produce what are called positive supercoils. And it's another way to rotate the DNA against its grain. You will produce negative supercoils. And those positive and negative supercoils are indiscernible at the level of a few base pairs. So when the enzymes bind there, the DNA will rotate into two different directions, but the enzyme itself is not centrally symmetrical with respect to the helical axis. So with SUMGOR, I offer you an analogy that involves pushing a piece of meat inside the meat grinder. The meat grinder, the DNA, has a helical coil, right? So it's one way if you twist the DNA of the meat grinder in one direction, the meat will respond in one way, and the meat will respond in the other way if you rotate the meat grinder in the opposite direction. In short, what the protein sees from the side of the DNA is different if you rotate positive and negative supercoiling. And this is the kind of insight that simulations can give, because up to the point when we started to do this, people thought that the positive and negative supercoils are released similarly by the enzyme. And the calculation of the free energy profile showed differently. I'll return to that. But let me first tell you what the experiments are about. What Ninke did was to attach magnetic beads at the end of the DNA and actually, just like in my example, supercoil the telephone cord. Well, here's to supercoil the DNA double strand. So twisting the magnet around, she was able to twist the bead, which in turn produced supercoils. And then the enzymes were released, and one at a time, the enzyme removed one supercoil after another. Each time a supercoil was removed, because there was tension upwards on the magnetic bead, a jolt was felt in the measurement apparatus. So in other words, the number of jolts is equal to the number of times the DNA has rotated, which is equal to the number of times the topoisomerase performed its enzymatic function. So divided by unit time, this will give you the rate of the kinetics involved in DNA supercoil relaxation, measured one molecule at a time. And so with kinetics in hand and the calculation of this free energy profile, we can compare simulation and experiment. Plotted here is the free energy profile on the ordinate versus the rotation angle on the abscissa. So you see here 0, 180, 360. This is a complete rotation. You see minima at multiples of 2 pi, and you see barriers at multiples of pi. This means that the DNA diffuses and jumps over the barriers, and it's going downward, which means that relaxation of the supercoil is facilitated. And the height of the barrier is going to dictate what the rate of the conformational transition for one supercoil removal is going to be. So we can estimate that rate from the calculation, and we can compare with the rate that's measured in the experiment. Okay? I won't talk about what the two curves are. You can also do this with an anti-cancer drug. If you can inhibit topoisomerase, you can stop DNA replication, so that has medical applications. Okay? So that's the result of supercoil removal. But that's only in the positive axis. In the negative axis, we have the relaxation of negative supercoils. And there you see that the barriers are different, and we can actually calculate those. And since the simulation has been done, there appeared an experimental measure in which the hypothesis that positive versus negative supercoils are different has been confirmed. So that was the story about how torque applied to the DNA can produce supercoils and how enzymes release those supercoils via a comparison of simulation and experiment. Next comes a story about the interaction between man-made nanoparticles, dendrimers, PAMAM dendrimers so-called, and condensed DNA. Here you see an AFM picture of what I would call naked DNA, means DNA. Of course, it's not really in solution, but you can think of it as that. That's how it would exist here. It's absorbed on the surface of some material so that it can be visualized. Here, it doesn't have too many nanoparticles. The nanoparticles are these white dots. I will tell you later what they are. If you keep increasing the concentration of those nanodots, the DNA will compact. So now it will look like this, which is in volume 1,000 times smaller than the volume of this object. So you have a very efficient way of compacting DNA. And in a compact state, this is useful to deliver it in a compact state if you want it to pass the lipid bilayer barrier. OK. This compact state of DNA can be measured experimentally with single molecule tweezers. So what you can do is you can grab the ends of this DNA and pull them apart at the same time as measure at different concentrations of the dendrimer. These dendrimers are branched particles like this. You have a central moiety that branches in two. That's generation 0. Those terminations branch in 2, so you have generation 1. The ends of generation 1 branch in 2, so you have generation 3, and so on and so forth. So you keep increasing the number of bifurcations at the end of each dendron, of each branch. And each time you do that, you increase by 1 generation. So here is what is measured in the experiment. Typically, force versus extension profiles are shown. So you see here that as I extend DNA, the force will increase. Then a curious force profile will be reached. Then the force will increase abruptly. That's in the presence of dendrimers. In the absence of dendrimers, the force starts to kick in at about 6 micrometers length of the DNA, when pretty much the entire DNA is pulled taut. And so what this slope is feeling the elasticity of completely taut DNA. But here, there's something else going on. There's a plateau. And if you are in my thermodynamics class right now, for the people in the back, we're talking about phase transition. Well, this is an example of a small thermodynamic system phase transition, because it involves the coexistence between a condensed phase and an extended phase. And this force plateau is pretty much like the pressure plateau that you have in pressure versus volume coordinates in typical discussions of the gas to liquid transition for real gases. So what happens is that you start with condensed DNA, you have a coexistence with extended DNA. Now all the DNA is extended and you pull it taut. Okay, now with this pressure versus volume diagram in mind, which is akin to the force extension diagram, let's go a little bit further. So the force versus extension diagram is very well described by this equation. This is force, F, versus extension, X. It's just a mathematical formula that involves a key parameter called the persistence length. You don't have to know what this is. It's the decay length of some tangent correlation function. It describes very well this force versus extension, the profile of naked DNA. But it does not describe the profile of the extension versus force in the case of dendrimer condensed DNA, and that's reminiscent of the ideal gas versus real gas situation. What did van der Waals need to do in order to get a flat pressure versus volume coexistence curve out of the ideal gas law? That's exactly what we will have to do to get out of the force extension curve modeled by the so-called worm-like chain model to get the real situation in the presence of dendrimers. What van der Waals did have to introduce were two parameters, A and B, that describes the microscopics of real gases, excluded volume and attraction. So similarly here, we'll have to do the microscopics, well, for us, mesoscopics, really, of the dendrimer DNA interaction, because now we have that interaction to describe. So that is done via molecular dynamics simulations. We compute the forces of interaction between the dendrimer and the DNA. And once we compute them, we can build a statistical mechanical model for the force versus extension that I won't go into detail, but that reproduces very well the experiment. It doesn't matter which one is which. Blue is the, for the sake of being exact, the blue is the experiment and the black is our simulation. Note here that there is no fit in the simulation. There is no fitting of parameters to get the experiment. The simulation gets directly the force extension curve via the calculation of the free energy as a function of the extension. This is supposed to be a quick walk, so I won't go into details. Okay, so what did we do? We simulated locally what happens with dendrimers, which are these black dots interacting with DNA, this thread here. And out of that, we extrapolated the macroscopic dynamics that leads to a force extension curve in the pooling apparatus. Okay, this is what we've done in this context. So now I will talk about the last topic involving what happens to DNA when it undergoes even more harsh contortions, and that will imply DNA squeezed inside the viral capsids. That will probably give me 5 more minutes to talk. But I'll need probably 5 more minutes to talk. Okay, so you probably know the details of it. In bacteriophages, you have, which are a certain kind of viruses, you have a capsid, you have a shell of proteins, and inside that shell, DNA is packed. It's packed at about 60 atmospheres. A good champagne is at about 6 atmospheres, so this is about 10 times the pressure in a champagne bottle. When DNA gets out of the champagne bottle, it does so through a neck, through a called portal pore, which is this reddish hole here. And that's also where it goes through when it's packed by a biomolecular motor that pushes it around and compacts it. But let's talk about the way it gets out first before talking about, instead of talking about how it gets in. This is a slice through a full viral head. You see that DNA is inside here squeezed at incredible compaction, and it has to get out when it undergoes infection, when it infects a cell, and so it gets out through this ring, red ring here, that's shown in detail in this slide. This is the DNA. This is a cut through the so-called portal pore, and this is a molecular dynamics setup for a simulation in which we will probe how DNA gets out of the viral capsid through this pore. The reason why we became interested in that was because there existed for a long time in the literature a model that had it that the DNA, when it gets out or in the portal, through the portal pore, it actually rotates like a corkscrew would when you push it in the cork. Okay, and that had various that was for various reasons, mainly having to do with a symmetry mismatch between the motor that pushes the DNA through and the six-fold, that was five-fold symmetric, and the portal was six-fold symmetric. So the symmetry mismatch led people to believe that DNA actually rotates like a corkscrew when it gets in and out of the DNA. When that was tested with single molecule first resonance spectroscopy with FRET, and two dyes were placed on what is effectively the DNA and what was effectively the portal pore, with the purpose of seeing a rotation. How would one see a rotation if one puts dyes? Well, if the corkscrew model is correct, right, and I place dyes on the DNA as it goes through, there will be a 360 rotation, so one dye will move relative to the other sufficiently that there will be a distance change that will lead to two colors in the FRET measurement. But that kind of color change was not seen. In other words, there was no large motion between the two dyes, which implied that apparently the DNA did not rotate. So that's what we set up to do. We put the DNA inside a cryo-EM structure that we fitted with a crystal structure of a monomeric protein that makes the 12-mer pore. And we did, guess what, calculated potentials of mean force for the interaction of DNA with the protein. Potentials of mean force, this time in 2 degrees of freedom. 1 degree of freedom involves the motion of DNA in and out without rotation, let's call this Z, and the other involves rotation of the DNA at constant Z inside the pore. So in other words, we have a potential of mean force in which energy goes up. This is the rotational degree of freedom. This is the translational degree of freedom. Okay, the free energy comes towards you in this plot, and when everything is said and done, after about 4 or 5 months of computing, we produce this egg rate potential, which has minima, maxima, and saddle points. And so if you want to predict, this is the central result of the simulation. This result will tell you, will lead to a model of how DNA goes through. If this is a free energy profile, and if this reaction coordinates are the slowest degrees of freedom, then the minimum free energy path along this profile will be how the system moves. Okay, so if you calculate the minimum free energy pathway, it will be like a connection between these 2 points. Right? I go from one minimum on the egg rate to another via a saddle point, which sits around here. That's the lowest free energy pathway. Well, that predicts at first sight rotational motion of the DNA, because look, this minimum free energy pathway is tilted with respect to the, it's not perpendicular to the rotation, meaning as it translates, the last slide went perhaps a little bit too quickly for you to understand, but you got the big picture, right? You remember that the minimum free energy pathway was tilted, right? Which involved the implication that the DNA will rotate, okay? And actually, it's good that I don't have the visual aid anymore. I will just use my laptop and imagine that my laptop is the egg rate potential, right? So I have minima in the egg rate, and this is rotation, and this is translation, right? So you saw that the minimum free energy path involves moving like this. But once you move like this, that's DNA rotating, right? It'll rotate, but when the virus is full, there will be something resisting this rotation, right? You will rotate corkscrew-wise against the resistance of a torque inside. But torque couples to angle, right? So while this was flat before, now because you apply torque, torque has the effect to tilt the landscape and raise the rotational degree of freedom. So what used to be minimum, now the ball that would roll in the accurate potential hops on your left. And that releases torque, so the free energy surface jumps back until, again, sufficient torque is built, and then the ball falls back. And this motion involves a little bit of rotation. Sufficient torque builds up. The force, the torque pushes back the energy landscape. So you have rotation, yet you don't. You have local rotation, but you don't have global rotation. In other words, the dies move a little bit relative to each other, and then they slip back. And that's why you don't see, in our interpretation, the full rotation of the DNA. And this picture is consistent with the measurement of no rotation, no major difference in the distance between the dies. Okay, so that's pretty much it, wanting or not. Thank you for your attention. What I did to recapitulate was to show local dynamics, DNA bases flipping out, DNA bases flipping over. Then I talked about the relaxation of supercoils, the compaction of DNA, and the ejection of DNA from viruses. Thank you for your attention. Don't worry about it.
It automatically turns off at 5 o'clock, and then it says locked out on the screen. Questions?
Yes, please.
So do you see the rotation and then until torque builds up until it relaxes? Do you see that for DNA entering and also exiting?
This discussion is pertinent for mostly for ejection. Okay, when the DNA is pushed inside, it is pushed by the so-called biomolecular machinery. These are either RNA-based or protein-based biomolecular motors that push it in. But the interaction between the DNA and the pore exists also when the motor pushes it in. We don't have there is no kinetic data on the import. Pushing in means import of the DNA, that we can compare it. So that's why I'm saying that it is more pertinent for ejection, for which there are kinetic measurements. In those cases, we know from experiments what is the rate of DNA ejection from a relatively full capsid. So in that case, we can compare the rate constant with the barrier height that we compute in the simulation. Right? It will depend exponentially on the barrier height. Right? The rate constant will depend exponentially on the barrier height, so we can, we actually put this in accord. So it's more pertinent for import, for ejection, not for import. Yes, Doug?
In the double helix, DNA is stabilized typically by interactions with ions, positive ions.
Yes.
Because of the large number of negative charges along the phosphate backbone. When you make the transition from B to P, do ions go away as the phosphates are?
Yeah, that's a good point. So the question is like this: the sugar phosphate backbone is highly negatively charged. It's -1 at each base. Okay. So in the bDNA form, the two phosphate backbones were relatively far apart. But here, they're squeezed in. So they will repel more than they did before, which is an additional unstabilizing effect. This is in part one of the reasons why the free energy of this is up in the absence of twist and torque. Now, what in a very different realm, when you have two double helical DNA molecules, they are negatively charged. This is negatively charged, so all they can do is to repel, you may think. However, if you put in multivalent positive ions in between the two negative DNAs, you can make the DNA molecules attract, the double helical molecules attract. Okay? And similarly here, what we have seen in the simulation, this time on the single-stranded DNA, we would see that monovalent ions would hop inside the two, from outside will hop inside and facilitate, somehow mediate the interaction between the single-stranded DNA. If that was artifactual or not and how long they live there, that we cannot say. But this I can offer in response.
So let's thank you, Juan, again.
Thank you.